When talking about DNA replication, we start with a diagram like this. Here we can see a single strand of DNA on the left-hand side, and it's being unwound into the two opposite strands. And on both of those sides, the top and the bottom, we see the complementary bases being attached onto each of these single strands of DNA. Now that works because the two strands were initially complementary, so if we have A here, T here, if there's a C here, there's a G here, and when we separate those rules are going to remain the same. So as long as we're matching up both of these single strands with their complementary bases, we're going to end up with two identical strands. The problem, however, or not so much problem, but the complicating factor here is the fact that DNA is not two parallel strands, but as two anti-parallel strands. And using my hands to visualize that, we've talked about this in class, but the palm of my hand would be the sugar, my fingers are the phosphate, my thumb is the nitrogen base. When we think of two nucleotides that are complementary to each other matching up, they're not like this. They're actually reversed like this, meaning that the orientation of the sugars and the phosphates and even the nitrogen base in the middle are opposite of each other. They're facing opposite directions, physically pointed in opposite directions. And that's significant because, as we've talked about throughout this course, there is such specificity to the shape of proteins and enzymes and molecules, and the way in which they interact and the chemistry that governs those attractions is so dependent upon the shape and orientation of the relative atoms and pieces that make up these molecules, that something like being flipped upside down actually means that now there's a fundamental different way in which these interact with different enzymes. And so what we see is we have one side where we have a protein called DNA polymerase III, which is responsible for matching up with the various nucleotides, new nucleotides, with the original strand. However, on the other side, we see sort of a whole bunch of events going on here, and that's to accommodate for the fact that this shape difference means that enzymes can travel in certain directions and not other directions on the strand of DNA. So we're going to walk through that process, and we'll bring into the conversation all of these. If you see here, though, this top strand goes from a 3' end, and if you follow it through, to a 5' end. So this top strand is 3 to 5 from right to left, and the bottom original strand is 5 to 3 from right to left. So again, anti-parallel. Let's assume that we've had helicase has unwound DNA. We have these stabilizing proteins. We won't talk a lot about those, but essentially they're protecting the single-stranded DNA, preventing it from rewinding, and just generally keeping stuff normal and preventing any sort of unfortunate damage or interaction with that single-stranded DNA. But that's happened now, and we're going to zoom in on the leading strand that has been separated. The first event that's going to occur on any instance of DNA replication is that an enzyme called primase is going to come along. It's going to match to a certain region of that single-stranded DNA, and it's going to leave behind a small piece of RNA primer. Now, this is RNA, which means it is chemically, physically different than DNA, has a different sugar, a different shape. And as this puts this down, what's going to be able to happen is that DNA polymerase III now has this sort of target of where to grab on and begin the process of replication. So this RNA primer is a way to signal and attract the DNA polymerase to attach. DNA polymerase wouldn't be able to just based off of its shape, wouldn't sort of know where to attach onto the DNA. Not that proteins know things, but we've talked about that. So when DNA polymerase III comes along, it's then going to begin moving down the length of the DNA, and it's able to do that as long as it is following a strand of DNA in the 3' to 5' direction. So polymerase follows 3 to 5. Now what it's creating is actually going to be an anti-parallel strand. So what it's creating is going to start as a 5' end here and be oriented towards the 3' end on the left. But simple fact to remember is that DNA polymerase III follows 3' to 5'. So that continues to happen. This is called the leading strand because that's it. It simply can go. And the DNA polymerase III would really get caught up to and be following just behind the helicase. So that way, as the DNA strand continues to unravel, polymerase III on the leading strand is just cruising along. The problem is that we just established that DNA polymerase III can follow DNA from 3 to 5. So on the other end, the lagging strand. On the lagging strand, we see, adjust myself there, we see this going from the 5' on the right-hand side to the 3' on the left-hand side. So the issue is that we can't just have a DNA polymerase cruising along to the left like we did above, because these molecules, if we looked at the actual shape of this strand, this single strand of DNA, we'd see that it's mirrored to the one that we saw before, that the molecules are actually pointed in opposite directions, and therefore they would not fit DNA polymerase being able to move from 5 to 3. So what happens is really the same story. A primase comes along and puts down a primer. Now it does that in the 3 to 5 direction. So here comes primase, and primase leaves behind a primer. When DNA polymerase is attracted to this primer, again, DNA polymerase can only go in a 3 to 5 direction. So DNA polymerase III comes along just like we saw before, except this time, even though we're unwinding to the left, it has to travel to the right. It has to follow the DNA strand from 3 to 5. And so it's able to do that, and it does so well, just like we saw before. But eventually it's going to hit a point that's either already been replicated or it's the end of the chromosome that is being replicated, and its job will be done. We call this the lagging strand because now what's happening is you have to imagine that off the screen to the left, more DNA is being unwound and prepared for replication. And so we're going to slide our diagram down. We're going to slide our diagram down, and now we're going to see a new primase come in and match up with the single-strand DNA and leave behind a primer. And another DNA polymerase III will grab onto that primer and will begin to replicate, doing its job matching up the complementary bases, complementary nucleotides. And it will do so until it bumps into that last primer that we saw. And at that point, it lets go. This will continue to happen, constantly lagging a little bit behind the unwinding, because the unwinding needs to happen. It needs to give enough room for the primase to come in, leave a primer, DNA polymerase comes in, and then goes until it hits where it's already been replicated. More unwinding, a primase comes in, DNA polymerase comes in, and we get it in these little chunks or fragments called Okazaki fragments. There's some additional actions that need to happen. Remember, meanwhile, on the leading strand, we're just cruising along. So on the lagging strand, we've got these little segments being made. Some additional actions need to happen. Looking at the screen right here, and even on the leading strand too, originally, we have some RNA that's in the midst of our DNA. Generally not good, right? We're trying to make an exact copy of our DNA. It can't really be a good exact copy if it's got little snippets of RNA throughout it. So one thing that happens right away is we have a different polymerase called DNA polymerase I that comes along, and all it does is it matches single-stranded DNA with its appropriate complementary nucleotides. But it does so while giving the boot to the RNA. So what happens here is the DNA polymerase replaces that RNA with DNA, as we saw. Now, we should notice there's a problem. The problem is that polymerase can't actually do everything. Polymerase has such a specific job. It matches single-stranded DNA to its complementary base. It brings in the complementary base, attaches it to whatever the previous nucleotide was in that new strand, but it can't attach it to the next thing. And so DNA polymerase I here, when it bumps into the end, it doesn't have the capability to attach these final nucleotides together, to do the dehydration synthesis to link those together. And so that's why we have another enzyme to fix this problem here, to fix these breaks in the backbone of our new strand, and that is called ligase. And ligase has a very simple job. Pops in, stitches that up, and then it goes. So ligase will follow along the process and basically fix the gaps in the phosphorus to sugar backbone of the DNA and correct any of those spots between these various segments on the lagging strand that need to be attached. So all of this is happening simultaneously. We see the leading strand, DNA polymerase III, just chugging along to the left. On the lagging strand, here's DNA polymerase III. It's moving to the right. In both cases, they're following their strands in the 3 to 5 direction. So there's consistency there. But because of this, we end up with lots of little segments. This segment got done, then this segment got done. More unwinding will happen, a new segment will happen, more unwinding, a new segment, until we're done the replication entirely. If I take this diagram and I make a copy of it and flip it, we can begin to understand how this actually happens a little bit more realistically within a cell. When we put them side by side, and then much more when we bring them together, this shows us what's called an ORI, or an ORI, or an origin of replication, which is a location within a strand, right in the middle of the strand, where we basically begin the replication process and we travel in both directions. So it's not just like we start at one end and work all the way to the other. It's hundreds of millions of nucleotides. That's crazy, right? So we start in the middle, can go both directions. Now you might say, well, doesn't that just cut the time down in half? That's true, except that in a strand of DNA, you're going to have countless ORIs all happening simultaneously, moving towards each other. And so with a whole swarm of polymerases and ligases and primases and all of these components working together simultaneously, an entire strand, an entire chromosome's worth of DNA can be replicated very, very quickly, because it's all happening multiple times at the same time, which is one of those sort of foundational chemistry tenets we've talked about with the cell. We like to talk about events as being these very calm, specific, orderly things, but at the end of the day, we have to remember it's chemical chaos, and all of it's happening on top of each other, all of it's happening at once, which begins to explain to us how we can make thousands of cells per minute within our bodies, replicating DNA for each one of those mitotic divisions. It has to be, first off, simple, based on simple rules like A's and T's matching and G's and C's matching. It has to be very small, right? We're on the 2 nanometer scale of the width of DNA, and it has to have that support of the enzymes that are facilitating all of that. So this is the process of replication. The leading and lagging strands, as we saw, and looking at this, we can see how this top part here on the top left is leading strand. This is our lagging strand, and it's opposite on the other side. Here on the bottom side is the leading strand, because if we're moving to the right, well, polymerase can move to the right on the bottom of these new strands. And likewise, up here, we see lagging. So when we look at leading and lagging, we want to have an understanding of why does that have to happen? And the simple answer is that polymerase follows 3 to 5 prime DNA. And once you understand that, the consequences of that, I think, become much more reasonable and logical, and we're able to think through this a lot more effectively, just with sort of understanding the basis by which all of it's happening. Awesome. Hope that is helpful.